Hello, everyone. My name is Sam Porter. I'm a postdoctoral research fellow in the Department of Microbiology and Immunology at the University of Michigan Medical School. And I'm here today to tell you about some of my research. This talk is entitled Building a Virus Factory, Investigating How RNA Virus Replication Organelles Form. So let's start with some background on what are flaviviruses, which is viruses that we study in my lab. Flaviviruses are a family of small, roughly 50 nanometer spherical viruses with a simple RNA genome. They are transmitted into humans via the bites of infected mosquitoes. Importantly for human health, there are no FDA-approved antiviral therapies, and there's only one FDA-approved vaccine which not all people are eligible for. So why do we care about flaviviruses? So flaviviruses are increasingly important in global health. The viruses we study in the lab include dengue virus, yellow fever virus, Zika virus, and West Nile virus. And collectively, they infect hundreds of millions of people every year, causing tens or hundreds of thousands of deaths. As you can see here by this map, most of these infections occur in areas in the global south, in the tropical warm regions where these mosquitoes like to live. However, in recent years, we've seen the emergence of West Nile virus all over the entire lower 48 United States. So as the climate continues to warm due to global climate change, the range in which these mosquitoes can breed is increasing northward every year. It inches northward by a little bit. And so in the near future, the two species of mosquitoes that carry, for example, dengue and Zika virus, Aedes aegypti and Aedes albopictus, will start to become more widespread across the entire southern United States. So this will become a threat into the United States as well. So I want to go over a brief summary of how flaviviruses replicate. The first step is they bind to a receptor on the cell surface. Then they begin to enter the cell where they get inside a vesicle. They then escape from that vessel and they go to something called the ER, which is this squiggly little thing down here as part of the cell. At the ER, they make their viral proteins and they form replication organelles, which is a critical thing I'll tell you more about in a second. In the replication organelles, you can make more progeny viruses, which then traffic to the cell surface and escape from the cell and go on to infect the next cell. So, what are replication organelles? You can think of these as virus factories where the virus can synthesize its proteins, replicate its genome, and assemble new virus particles. And what these physically are structures called invaginations in the ER membrane. Essentially, this is the ER membrane, and there is a dent that's formed and it slowly makes this bubble-like compartment where all these activities take place. And so what is the purpose? Why does the virus bother making these structures? The first one is to protect the virus. So these replication organelles physically hide the virus from sensors in the cell that can detect replicating genomes and initiate an antiviral response. Obviously, the virus doesn't want that. The other thing it serves to do is to organize replication components. So you can think of this when you're cooking in the kitchen. You want to have all your little stuff, all your tools and your ingredients right next to you. And so that's what this does. That's a scaffolding physically to concentrate things closer together. And so it's easier for the virus to do the things that it needs to do to replicate. So what we're interested in is investigating what parts these replication organelles are made of. We know a fair amount about the viral proteins that are synthesized by the virus inside infected cells that help to contribute to the formation of the structures. You can see some of these proteins here, NS1, NS2A, NS2B, NS4A, and NS4B. But what we don't know much about is what cellular proteins are involved in the formation of these organelles. We know there must be some cellular proteins that help to make the structure, but we don't exactly know which ones are involved. And finding out is relatively complicated. So how do we figure out what cellular genes are involved in the replication organelle formation? Well, what we do is we start with a plate of cells in which every single gene in the human genome has been knocked out with a technique called CRISPR. And so what we can do is we can give these cells a lethal dose of dengue virus, let them grow for a while, and then what will happen is that most of the cells are going to die. The cells that survive are resistant to dengue virus infection. And we can take those cells, sequence their DNA, and find out what genes were knocked out. And that tells us that that gene that was knocked out is important for the virus's replication. So when we do this experiment, one of the strongest genes that we find are a series of genes involved in the complex called the ER membrane complex. This is a multi-protein subunit complex that is involved in the stabilization of certain proteins. And so what we think is happening here is that this complex called the EMC is helping to synthesize viral proteins, which are then involved in the formation of replication organelles. So when this is happening properly, we get full replication organelle formation, and it goes on to make progeny viruses, and the cell dies. When the EMC is deleted, such as in our experiments, these proteins cannot be folded properly. The replication never occurs, and the cell actually survives the infection.